Namaskar. Today we'll study about exons and introns. And what is this exon and intron? The gene has two regions, exon and intron. And what is their role? As we know, DNA is the source of your hereditary information, and DNA has the information which protein can be synthesized. So first, this double helical structure of DNA by transcription is converted to messenger RNA. Now messenger RNA has the information. Then this messenger RNA has this bases, and we call them three bases together, we call them as code. Suppose in the messenger RNA the code is say AUG. So this is the code, and the transfer RNA at one end it has the amino acid and the other end it has the anticodon. So A, U for A, G for C. So UAC, the tRNA with UAC anticodon and corresponding amino acid will recognize this AUG in the chain of mRNA and will provide this amino acid. So one after another the amino acids will be provided, and this is process of translation, and we get the protein. But this coding region is only present in this exon, not in the intron. So this intron is present in between two exons, and when there is transcription we get this RNA where both exons and introns are there. But since this intron is the non-coding region, and if you see the difference, the exons name is called exons because it is expressed, it has the coding sequences, it is the part of gene where the sequence, the part of gene sequence which are expressed. So it is found in both eukaryotes as well as prokaryotes. But introns, they are not expressed and come in between exons, and this is the portion of gene that does not code for amino acids. So we can call them as non-coding sequences, and it is found in eukaryotes only. So in prokaryotes there is only exon, whereas in eukaryotes we have both introns and exons. So in a human cell there is both introns and exons, but in a bacteria there is only exons. So only coding sequence. So there is no intron in case of bacteria. But in our cell, in human cell, there is both intron and exon, and since intron is the non-coding sequence, we have to remove it. So in a human gene, there is average 8.8 exons. So you can say 9 exons are there, and in between exons, since introns are there, so average 7.8 or approximately 8 introns per gene are there. So number of genes in a human genome is around 26,564. So total exons is 2,33,785. Total introns is 2,07,344. So average, if you calculate, it will come around 8.8 exons per gene and 7.8 introns per gene. So this gene is having both exon and intron, and after transcription also this RNA, this transcript also contains both exon and intron, and this intron has to be removed because this is the this has the non-coding sequence. So coding sequence is only available with exon, and the process which removes this intron, that is called the splicing. So mRNA splicing is the process where the introns are removed and only exons will be available for translation process. So we after splicing we call it as the processed messenger RNA. Up to this it happens inside the nucleus, then it will, through the nuclear pore, it will come out into the cytoplasm for protein synthesis. And if you see, this messenger RNA is having 300 to 3000 nucleotides. So as we know, every 3 nucleotides code for one amino acid. So we can have 100 to 1000 amino acids. So this amino acid chain will, after some post translational modification, will lead to the protein. So we have proteins whose amino acid chain length will vary from 100 to 1000 because of this. So mRNA splicing is the process which removes the intron, and there will be exons and which can code, and we can have the proteins. Okay. Then what are the applications? One application is this alternative splicing. What is alternative splicing? So after mRNA splicing, suppose here say we have 1, 2, 3, 4 exons are there. So say E1, E2, E3, E4. Then we can have a combination where E1 will be followed by E2, followed by E3, followed by E4. So say this is E1, this is E2, this is E3, this is E4. This is one combination. We can have another combination E2 followed by E3, followed by E1, followed by E4. We can have another combination E3 followed by E2 followed by E4 followed by E1. So different combinations we can have. Suppose here we have 3 exons. So E1, E3, E2, one combination. E2, E1, E3, another combination. E3, E2, E1, another combination. So if different combinations we can get from the same gene, then we will get different proteins from the same gene. So when we get different proteins from same gene because exons joining in different combinations, we call this process as alternative splicing. So one gene can lead to synthesize different proteins because of mRNA splicing and joining of exons in different combinations. This is called alternative splicing. But when this recombination between exons of different genes occurs after mRNA splicing, if exons from different genes, so suppose one exon from say one gene and then another exon from another gene, so that will provide us a novel gene, a new gene, and that is the basis of evolution. And because of this is the mechanism of evolution, and we call this process as exon shuffling. So this exon shuffling is a process where recombination between exons of different genes occur, so that we get novel genes, new genes, and that is the basis of evolution. So this is all about the exons and introns. Thank you.